Welcome to Eye on the Cure, a podcast from the Foundation Fighting Blindness, providing science research, news, and insights about the world of vision and blinding diseases.
Welcome, everyone, to the Eye on the Cure podcast. I'm your host, Ben Shaberman with the Foundation Fighting Blindness, and I'm excited to have a conversation with Dr. Lance Baldo. He's the CEO of Beacon Therapeutics, which has fully enrolled the phase 2/3 VISTA clinical trial for its X-linked retinitis pigmentosa gene therapy, which is targeting people with mutations and RPGR. We're very excited about this trial. And Lance, we're very excited to have you on the podcast. Welcome.
Ben, I'm really excited to be here and just to be able to join a list of, really prominent people that you've had in the past. So it's absolutely my privilege, Ben.
Well, you're an addition to that list of prominent guests. So thanks for being here. And a little more on Lance. He has more than 20 years of leadership experience in biopharmaceuticals, including the successful launch of 2 new indications and a new formulation for Lucentis while he was at Genentech. And for those of you that don't know, Lucentis was first developed for wet AMD. Anyway, prior to joining Beacon, he served as chief medical officer at Freenome, which is an early cancer detection company. And there he led the design and execution of the company's medical strategy to support its pipeline from clinical trials through registration and commercialization. And Lance also served as chief medical officer at Adaptive Biotechnologies, an immune medicines company. And there he helped to lead the transition from a private to publicly traded company. And way back when, he earned his medical degree from the University of Connecticut School of Medicine and a Bachelor of Arts in Biology from Johns Hopkins. And I'm curious, Lance, to start off our conversation, you got a medical degree, an MD, and that involved a lot of clinical training, of course, but you went directly into industry and therapy development. What inspired you to take that track?
First, Ben, you're making me feel old when I think back now that medical degree was gained over 30 years ago now. I think some of those textbooks had been rewritten for better for worse.
Well, I won't try to do the math to figure out your age, but that is a little while ago.
Yeah, no, I loved being a doctor, and I love being a doctor. I think it's a really amazing profession, and the impact that you can have on people and society. And Ben, I never really chose to leave medicine. It just so happened that when I was doing my surgical training as a young resident in orthopedic surgery, actually, I had an idea for a company. And that company, the whole thesis was that physicians were going to be mobile and they're going to be moving around hospitals and clinics and things like that. And so what that company did, again, way back in the 90s, is we mobilized medical information and put it onto mobile devices. At that time, it was a Palm Pilot. That's how much I'm dating myself. You think back now, 30 years later, and it's obvious that physicians have everything at their fingertips. But in the late 90s, it was not so obvious. And after founding that company and running that company for a number of years, Ben, I just decided that I kind of got the business bug. And as much as I knew I would miss treating patients directly and being in the operating room, I decided that I was going to devote my career to ways that I could help patients in different ways. And specifically, I moved from there to pharmaceuticals, Ben.
Well, we're very glad you chose the therapy development route because you've done some great work, and we're excited, obviously, to have you at Beacon. But reading your background, you've been involved in cancer, the immune space, and then you did eventually move into ophthalmology. What inspired you to get involved in ophthalmology, especially this time around with Beacon?
Yeah, I'm a bit of a storyteller, Ben, so I'll give the story on that. And, for better or for worse, I consider myself to be a bit of a generalist across the spectrum of kind of pharmaceuticals and biotechnology, having done a little bit of many things. But I was very fortunately sometime I'd say that was around 2015, I was working as a general manager in a Roche affiliate. I was the head of the New Zealand office for Roche. And I got a phone call from my boss who had spoken to the CEO of Genentech, and they needed a franchise head, so a person to lead ophthalmology for Genentech. And that would be a huge step up in my career, and I really felt humbled at the opportunity. But I had never worked in ophthalmology before, and so I had a little bit of this pit in my stomach, and I did a little bit of research to figure out like what would I need to learn and what would I need to know. And also, one of the things I was told at that time is you're going to love this community, the ophthalmology community, the retina community, incredibly great people, really focused on making a difference in patients and cures and things like that. And they weren't wrong. And so I was able to relocate to the Bay Area where I reside now and take over as the franchise head for ophthalmology. And it was a really steep learning curve trying to move from all these other areas I worked in to learn ophthalmology and specifically learn retina. But I was supported at Genentech by an amazing team, and also just being able to engage with external experts and clinicians really gave me the opportunity to learn and eventually love ophthalmology. I consider that to be, one of my favorite, if not the favorite specialty that I've had the privilege to work in, Ben. It's really just an amazing place with incredible innovations and more on the horizon. We're going to talk about one of them later, I know.
Certainly, certainly. And I'm thinking back to those early days in Lucentis development. As I read in your bio, you were involved in Lucentis development. And I remember very clearly, I was still relatively new at the foundation, but it was June 2006 when Lucentis was FDA approved. And that was such a game changer for wet AMD because there really wasn't much else that was working. Were you with Genentech at that point?
I didn't join Roche Genentech until 2010. So that was 4 years after I joined. And so I look at it as being given the opportunity to stand on the shoulders of giants. The work that I did, Ben, was helping to just take that initial indication and then move into other indications, including things like diabetic macular edema. We advanced the pre-filled syringe. So we took that great innovation that was Lucentis and then build onto it for over time while I was at the helm running commercial for Genentech.
Got it. Got it. So more recently, you took on the CEO role at Beacon. And obviously, to be a CEO, that's a pretty big leadership role. And then Beacon is in the rare retinal disease space. So what inspired you to get or to take on the role at Beacon and to be the CEO, which is obviously, again, a big responsibility?
Ben, I had CEO-like jobs when I was working for a large company like Roche Genentech. I mentioned I had run New Zealand, the country, as a general manager. I had been a life cycle leader, which are sometimes called the mini brand CEOs. And I just knew that that type of opportunity to have really broad impact on a company or program was really appealing to me. And so about 2 years ago, I made the decision that I was going to transition from the last two CMO roles and try to pursue a CEO job. And because I loved ophthalmology and retina so much, I was kind of hoping I was going to be able to narrow that to ophthalmology alone. And I was fortunate that two different job opportunities came my way. One of them was Beacon, Ben, and I pinched myself then, and I still pinch myself now for what a good fit it is for me. I'm a physician scientist, and I love and late stage drug developer. I love products that are in the clinic, and I love products that are close to helping patients. And at Beacon, we now have a product in laru-zova that we'll talk about, I'm sure, later, that could potentially just be a couple years away from helping thousands of patients across the world. And to have that opportunity to help the company navigate through the clinical work, the regulatory work, and the commercialization, it's an absolute privilege, but also some pressure, Ben. I'd say I feel, and I know my team feels, a lot of pressure because we don't have the opportunity to make missteps. If we make missteps, patients and their families are going to suffer. So that responsibility is not lost on me or anyone in the company, but especially me as the CEO and the ultimate person accountable for the work that we do.
And I can relate to the pressure, maybe not as a CEO of a company, because I'm not, of course, but blinding retinal diseases are progressive. The community feels such an urgency to get treatments across the finish line, whether obviously if you're an affected patient or a family member, you feel the urgency. And those of us at the foundation who are funding a lot of research, we feel the urgency. With all that said, science, whether it's lab science or in clinical trials, it just doesn't move that fast. And yeah, you definitely have the pressure of ensuring that the clinical trial goes as smoothly and successfully as possible. I can't imagine what that pressure is like.
Yeah, and fortunately, like any good team or any good company, I don't have to do it alone. And we had the privilege recently of Dr. Dan Chung joining us as our chief medical officer. And I know he's probably well known to many of your listeners, and I know he's well known to FFB, and he's been a great addition to the team. And so we're hoping that he's going to really help us steward this product and this company into the future as well, Ben.
Right, right. Yes, we know Dan very well from his days at Spark when Luxturna got FDA approved, then at Sparing Vision, which is a company we're invested in. And Dan has done a lot of great work and made some important innovations like Maze, the mobility course that was used to get approval for Luxturna. So you definitely have a good soldier there. So congratulations on that. And I do want to talk about your X-linked RP gene therapy in just a second. I want to give our listeners a little history on that gene therapy because an important moment in the development of that gene therapy came back in 2012 when Gus Aguirre and William Beltran at the University of Pennsylvania showed that an X-linked RP gene therapy actually worked pretty well in a canine model of XLRP. So that was 2012. They did additional studies to evaluate it at different stages of disease, and I'm sure there were other refinements. And then 6 years later, the company Applied Genetics Technology Corporation, or AGTC, which many of our longtime listeners probably recall, they actually launched the original phase 1/2 trial for that gene therapy. There were a couple of additional trials, sort of extension trials launched after that. And then finally, after AGTC went through some changes in its business structure and became ultimately Beacon, Beacon finally launched the phase 3, the VISTA trial, about a year ago? Year and a half ago?
It enrolled over 14 months. It enrolled over 14 months. Ben.
Okay. And excitingly, I'm going to steal a little of your thunder here. I know you recently reported that it's fully enrolled, which is awesome. Can you tell us more about this X-linked RP gene therapy?
Yeah. So the product used to be known as AGTC 501 because, as you rightly point out, it had been being developed by the AGTC labs. We've now, I'll say rebranded, but it's not technically branded. We've now adopted a derivation of the so-called scientific name, or the INN, and we call it laru-zova. So you'll be hearing me refer to this product as laru-zova. And what laru-zova does is able to localize to the photoreceptor cells in the back of the eye. And when it's there, it's able to allow those cells to produce the protein that's aberrant or missing within RPGR mutated XLRP and allow those photoreceptors to start to behave more normally. And when that happens, what we're seeing in some of the earlier clinical work that you alluded to, Ben, is we're seeing improvements in markers of visual function, things like microperimetry, FST, and really importantly, low luminance visual acuity. And I hope we have more time to talk about that later. And as you rightly pointed out, the key study, the pivotal study, is called VISTA. That is a study that was conducted in the US, Australia, and the UK. It was intended to enroll 75 patients, but fortunately we were able to over-enroll to 85 patients. And just this past June, it's fully enrolled. That study has a 12-month primary endpoint. And so before the end of next year, before the end of 2026, we will know what kind of an effect that medicine is going to have on these really, really important subjects that chose to be involved in this study and help to advance the science of X-linked retinitis pigmentosa. So couldn't be happier to have that study fully enrolled. In any indication, it's no small feat to fully enroll a study, and particularly when you're talking about a devastating rare disease like X-linked retinitis pigmentosa.
Certainly. And I had the honor and the privilege of moderating a gene therapy session at an FFB conference called the Investing in Cure Summit. Really was in the spring, late winter. And you presented results from a study called Dawn, which is, I guess, still ongoing, but there were some really nice results from that. It's a phase 2 study for that gene therapy. Can you talk about Dawn a little bit?
It's one of my favorite things to do, to talk about Dawn. First and foremost, it is a safety study. So the primary thing we wanted to do is make sure that this gene therapy is safe when administered in the contralateral eye, so the other eye, but also over the period and tracking safety over many years. But Ben, we did embed into the study a number of really important markers that we were interested in seeing what kind of an effect laru-zova would have on those markers, and very specifically low luminance visual acuity, and also mean microperimetry. And the entire technical term would be mean change from baseline and mean microperimetry. So how much does the back of the eye improve in terms of how it picks up light sensitivity? And those data are continuing to mature. When we present them most recently, we presented an update at Retina in Paris just a couple of weeks ago. A really high level summary is that we are seeing very significant improvements in subjects that are getting 2 and even 3 lines, not letters, lines of gains compared to where they were at baseline. So really rough numbers, and the numbers change over time as the data mature, but 60 to 70% of subjects getting 2 lines of gain, and somewhere between 30 to 40% getting 3 lines of gain. So these are really profound and important improvements that we're seeing in this Dawn study right now. We will continue to follow these patients. Number one, just to see how many more could hopefully improve, but also to look at the durability of these therapies as well, to make sure that what we're seeing at 12 months will also hold at 18, 24, 36, etc. And so we're just generally pretty excited about these data. They will serve as part of the ultimate filing package that we'll be putting in front of the European as well as the US regulators. And one other thing I'd say about this study, Ben, and you had mentioned, Dan and some of his work on maze. We, and my predecessors, I can't take credit for this, had some concerns about the maze being a discriminatory endpoint in XLRP. And so we knew this drug was doing, or had a strong suspicion this drug was doing something important in these patients at the back of the eye. But the question is, how could we measure that? And we felt like, and again, these were my predecessors, not myself, felt like low luminance visual acuity could be a really important discriminatory endpoint. And Dawn is really the first time that we looked at that prospectively and were able to prove to ourselves that we could have a meaningful impact on this really important marker of clinical improvement, Ben.
Yeah, I think that's a really important point. The maze that Dan developed or helped develop was the perfect endpoint for LCA, labor congenital amaurosis caused by RPE65 mutations, for the gene therapy for that. But X-linked RP is not LCA, and you really have to figure out what the optimal endpoint is for each condition and therapy. And that is no small task. And I know we're going to come back to low luminance visual acuity in a moment, but I want to ask you about Johnson & Johnson. And about the same time that meeting took place where you were in the session I was moderating, J&J also reported on their X-linked RP gene therapy, the results from their phase 3 clinical trial. And while it did improve vision for many people, they missed the endpoint, the primary endpoint that they had agreed upon with the FDA. And now we're not sure what's going to happen with their X-linked RP gene therapy. So seeing those results from J&J, how are you leveraging what they learned in the clinical trial for your gene therapy, or how does that inform your outlook for your clinical trial, if I can ask that?
Certainly, Ben. You learn a lot from studies, whether they're successful, whether they're unsuccessful in achieving their primary aims. And I guess first and foremost, I want to thank J&J and Mira for the ambition they had in setting up that study and for all the subjects, patients, families, and clinicians that were involved in that program. Because the learnings from that program are really helping to advance the entire field. And we've taken some of those learnings to the best we can, incorporate them in the work that we do and in the strategy that we employ to make sure that we can learn from some of the things that were done well and learn from some of the things that potentially could have been done better in terms of how we design our study and how we execute our statistical analysis plan. And as you're alluding to, while they failed on the mobility assessment or the so-called maze, there were some trends noted in secondary endpoints, including endpoints of microperimetry and low luminance visual acuity, although they weren't quite on the magnitude that we're seeing based on laru-zova in the Dawn study, but still comforting to see those trends that, if you do augment or if you do replace that protein, it seems to have some kind of an impact on our ability to improve these functional measures of vision. And we had already decided before that study, but it really reinforces our decision to have low luminance visual acuity as the primary endpoint, particularly for the US, within the VISTA study. And so for patients, at least, while a negative study is never a good thing, we're going to learn from that experience, and then we're going to move forward at pace to make sure that we can get laru-zova to all these patients that are waiting for a therapy to be able to help treat their condition.
Right, right. And what's interesting, and you did say this, is it really provided some affirmation for your selection of low luminance visual acuity as your primary endpoint, because that actually ended up being a pretty good endpoint for the J&J trial. So that's definitely a good thing. I'm wondering if you are able to talk about any differences in the actual gene therapy between, well, your gene therapy and what J&J developed, because on the surface, a viral gene therapy for X-linked RPGR mutations sounds like it would be pretty similar. There are differences, and I don't know if you're able to talk about those differences and again, and what you did versus what J&J did.
Yeah, and I certainly can, Ben. I would say there's some similarities, as you point out. I mean, these are AAV vector gene therapies administered subretinally, so put into the back of the eye. So that all is quite similar between the two. There's one key difference, and it's philosophically one of the reasons why I decided to join Beacon and felt like even had the J&J product succeeded, that we could have had a at least as good and maybe even better product, is that laru-zova replaces, so the transgene codes for the full-length RPGR protein, and that includes a really important part of the gene, which is called open reading frame 15, where about 40-50% of the mutations in RPGR XLRP are located. And so when laru-zova is administered, the full-length protein is then generated. And it was my belief and our belief that would tend to lead to better outcomes. Now, the clinical study has to speak, Ben, right? We could talk about philosophy and mechanisms all we want, but ultimately this has to read out in the context of the clinical study. But I think we're kind of seeing that play out right now, with the trends that we're seeing in the J&J study and the data that we're seeing with Dawn, that perhaps replacing that full-length protein is going to be important for the science, and it's going to be important for patients overall.
Right. I think that's a really good point to make. And for our listeners, we often talk about gene therapy as like gene replacement. You're putting in a new gene to replace the defective or mutated gene, which you are doing. But at the end of the day, it's how is that protein performing? Is the protein close to the protein that it should be, or it normally would be if the gene wasn't mutated? And at the end of the day, it's all about the protein. So it's exciting that you are helping to express a full-length protein, and hopefully that translates in vision improvements. So we'll see in the phase 3. No pun intended, of course. Let's move on to the rest of your pipeline, because you do have other things in development. Can you talk about any of those?
And we certainly can. The most advanced program after laru-zova that we're currently looking at is another gene therapy, which we call right now BTX-001. And that's a gene therapy that's administered intravitreally for the treatment of dry AMD. And for your listeners, I don't even need to go into the condition itself. It's pretty widely understood and has huge individual and societal impacts. And we're fortunate there are at least now two in the US therapies, not gene therapies, but therapies approved for that condition, but they don't reverse the course of disease. They just slow progression, which is still meaningful, but patients don't get positive feedback. And the treatment burden tends to be pretty high because they have to come back once every 2 months to get yet another injection in their eye. And then they're not feeling better. In other words, they're not going to their physician saying, thanks, doc. Now I can see better because I've been getting these shots every month or every 2 months. So the hypothesis is with BTX-001 is that we're going to try a one and done gene therapy where you administer the gene therapy intravitreally. We've now disclosed the target. It's a protein inhibitor of C5. And then it basically allows the cells of your eye, the choroid cells of your eye, to produce levels of this C5 inhibitor. So this way you can hopefully for a long time and maybe even indefinitely tamp down, some of the inflammation that's associated with dry AMD. That program right now is in primates. It's being studied in a GLP tox study. The plan is to open up the so-called IND or investigational new drug application by the end of the year and be in the clinic, meaning being in patients, being tested by the second half of next year. And so we're just trying to advance that program as well and as quickly as we can, Ben.
That's great. That's very exciting. And I know the dry AMD patients out there are definitely looking for more effective and less burdensome approaches to slowing, hopefully, the progression of their disease. And can you just talk a little bit deeper about how this gene therapy works and the role of inflammation in age-related macular degeneration?
Yeah, it's interesting. Many of these diseases, I may even go so far as to say most, have some type of an inflammatory component to them. And often it's when that inflammation kind of gets out of control and can't be controlled by the body's natural checkpoints. And there is something within, frankly, all over the body, but particularly within different parts of the eye, including the vitreous humor, called the complement system. And that complement system is involved in modulating some of the processes involved in inflammation and ultimately with something called apoptosis or programmed cell death. And so what we're trying, it is a complex system, and there's cleavage products and there's multiple different parts of the complement system. So we're trying to just figure out which parts of that complement system can we modulate or can we block that would allow us to prevent some of the downstream effects, which ultimately lead to death of a lot of the cells in the back of the eye that you need to have good vision, or sometimes the buildup of products in the back of the eye or in the retinal space that are going to impact your vision as well. And so that's what we're targeting here. And you made a really important point, Ben. Some of this is about incremental progress. How can we do better than what we have today? But we need to keep working on this. I'm a big believer in BTX-001, and I think it's going to make a difference for patients should we be able to develop it in the way that we want to. But all of these products are just a step in the direction of hopefully eventually being able to fully understand this disease, eradicate it, or maybe even reverse course. And I mean, that's the holy grail. But the only way that you can get there is by making incremental steps. And we believe that BTX-001 is a incremental step. It might be a large incremental step, but it's an incremental step towards making better medicines for these patients suffering from dry AMD.
Definitely, definitely. And we're excited to see that trial launch. And as I think about it, 2026 is going to be a big year for you. You're going to launch the dry AMD gene therapy trial, and we're going to hear about your X-linked RP gene therapy. So fingers crossed for 2026. In the meantime, it's been a wonderful conversation. I appreciate you taking time on this Friday afternoon. This is when we're recording. This will probably air in a week or so. But anyway, it's been a real pleasure talking to you. You really have done a wonderful job explaining your role, the current status and history of your different gene therapies. And again, Beacon is at a very exciting place, and it's fun to talk about it right now. And fingers crossed that things keep moving forward in such an impressive way.
This is an energizing way to end my week, Ben, and I appreciate you for it. Love to come back next year as we get more news. And just a thank you to FFB and the work that you do as general. I've worked across many therapy areas, and I have to say, as far as supporting patients and science, FFB may be one of the best, if not the best organizations I've ever had the privilege to work around. So thank you for all the work that you do, Ben.
Well, thank you for saying that. It's wonderful to have Beacon as a partner. And thanks again for being a speaker at some of our conferences. You do such a good job presenting the research. So thank you for that. And listeners, thank you as always for joining Eye on the Cure. It's great to have you, and we look forward to having you back for the next episode.
This has been the Eye on the Cure podcast. To make an impact, donate now at fightingblindness.org.